Okay, dear colleagues, welcome to our webinar. We are from Vilnius University Life Sciences Center, and we are present Instruct-ERIC, Lithuania. And I will start with a brief introduction of our Life Sciences Center, which was established around 8 years ago when three research institutes from Vilnius University, so Institute of Biochemistry, Institute of Biosciences, and Institute of Biotechnology, moved into a new building at the Vilnius University new campus. A few years later, the EMBL Partnership Institute on Gene Editing Technologies was also established at LSC. Now, the research at our Life Sciences Center covers a broad range of various areas, starting from biodiversity and ecosystems, and then with genome and epigenome editing, biomolecular structure and function. One of the most important topics studied here is bacterial and viral defense. So these days, we have the best known antiviral defense systems in bacteria is CRISPR-Cas, and of course it's directly related to genome engineering, but also we use various biochemical and structural methods to study other bacterial antiviral defense systems. So here at LSC, we perform X-ray crystallography, and we have equipment for automated crystal growing and inspection. And a few years ago, more than 3 years ago, in fact, then our local Cryo-EM facility was established. So we have a Giedrius Cryo-EM and all equipment required for sample preparation and data processing here at LSC. And we perform single particle analysis. And this brings me to my own presentation. So please feel free to type in questions into the Q&A section. After each presentation, we'll have a few minutes for answering some of the questions. So I'm Giedrius Sasnauskas, and I will talk about Cryo-EM studies of Cas12 family in the nucleases. Now, Cas12 enzymes, CRISPR-Cas nucleases, as well as CRISPR-Cas9 enzymes, are well best known for their genome editing applications. But their natural function is to protect bacteria from invading bacteriophages by cutting this invading DNA. Now, from this clans clustering, we can appreciate that Cas12 nucleases they are from a very large and diverse group of enzymes. In fact, most of the Latin alphabet is already used to name various subtypes of these Cas12 nucleases. Some of them are, for example, Cas12As, are well-known genome editors. Also note that the central part of this clans clustering is occupied by not a CRISPR-Cas nuclease, but enzymes called TnpBs that are also related to Cas12. But these are not CRISPR-Cas enzymes, but these come from bacterial transposons. Another group of related enzymes are fanzers that were discovered recently, and these are found in eukaryotic cells, not human cells, but in some fungi and some eukaryotic viruses. And today I will speak mostly on the TnpB from Deinococcus radiodurans here, and also mention some properties of some closely related Cas12 nucleases, for example, Cas12M, here the group at the top, or Cas12F, the yellow small crest. Now, where does this TnpB comes from? So it comes from a bacterial transposon. This transposon is surrounded by left-hand and right-hand sequences, shown here in green and blue. It is always inserted next to a short DNA motif called TAM, shown here in violet, a TT-GAT sequence in this particular case. And these transposons typically encode two proteins, TnpA transposase and TnpB. So the role and the mechanism of this TnpA transposase it's quite well understood for quite a long time, whereas TnpB for a long time its role remained elusive. Only a few years ago, our colleagues from LSC, led by Thoeris, they demonstrated that TnpB, which is a 400 amino acid protein, forms a complex with RNA transcript called right-hand derived RNA, or another name is omega RNA, which are transferred from the right end of this transposon. And this transcript also includes a fragment of downstream DNA shown here in red. This complex is around 100 kilodalton in size, 50% protein, 50% RNA. This is a nuclease that recognizes and cleaves DNA in the following targets. So target is TAM sequence, which is recognized by the protein, followed immediately by this red downstream DNA, which is recognized by this RNA transcript. And by changing sequence of this RNA transcript, it was possible to target TnpB to different DNA sequences. And it was also demonstrated that TnpB is capable of genome editing in eukaryotic cells. So, in fact, the smallest genome editor known. I would also like to point out that the natural function of TnpB is not to protect bacteria from viruses like Cas12 or Cas9 enzyme, so that it is a transposon enzyme, so it must help transposons somehow. So how does it do this? So excision of these transposons by TnpE usually happens during DNA replication, and these transposons are excised in single strand form. And when replication is over, only one of the newly synthesized DNA strand copies retains the transposon in its original location. And if cell divides at this moment, only one of the daughter cells would retain the transposon in its original location. But now that excision of this transposon, it brings together TAM and its downstream DNA sequences, in fact, generating a TnpB cleavage target. So TnpB presumably cuts this newly generated target, introducing a double-strand break, and repair this double-strand break via homologous recombination, reinserts the transposon into its original location. And if the cells divides at this moment, so of course both daughter cells have these transposon copies. Now, first we use Cryo-EM to solve the structure of the binary TnpB RNA complex. And what have we learned from this structure? So ironically, not much about the protein itself, because even before the AlphaFold, we had quite accurate model of this protein. But we learned a lot about the RNA. In fact, this RNA transcript, approximately 150 nucleotides, forms a quite a peculiar and complex structure that includes a pseudo-knot, a triplex region, stem 2, and a very long stem 1, and forms various contacts with the N-terminal REC and VET subdomains of this protein, and of course with the catalytic green REC domain. But of course, the most interesting thing was how this TnpB RNA complex would recognize the DNA and cleave it. So of course, we tried to solve the structure of the ternary complex, TnpB, RNA, and DNA. We use sort of a minimal DNA substrate reminiscent of its cleavage product. So what happened? We faced the preferred orientation problem, well-known problem in Cryo-EM and single particle analysis in Cryo-EM. We were not able to obtain interpretable map. So address this problem in such a way that we designed a so-called dual-end DNA substrate, where we in fact fused together two copies of this smaller substrate in head-to-head orientation. And we believe that this would allow binding of two TnpB molecules that are opposite end of this dual-end DNA. And by changing the distance between these sites, I expected that we would change the relative orientation between these two TnpB molecules, forcing some extra orientation between these two bound TnpB molecules. And in fact, after some optimization of this distance, we obtained an interpretable map. It was not perfect, but it was interpretable. We were able to model the structure here at the bottom. So we were able to model the DNA bound to this TnpB and RNA. So in this case, we see the DNA, including the time sequence at the top. And if we focus on this area where this is the bound DNA and look at all the contacts, we observe the things that one would expect In fact, we see a number of base-specific contacts made by the wet and rec subdomains to the TAM sequence. So this way, the protein directly recognizes the TAM sequence. It is reminiscent of PAM recognition in Cas12 and Cas9 enzymes. And also, it makes a number of these non-specific contacts to the DNA and RNA backbone in this heteroduplex region. So, and look at this heteroduplex real structure, so it's essentially immersed into the protein. Protein makes really quite a number of contacts to this DNA backbone and RNA, and we believe that these contacts help to test the quality of the heteroduplex to ensure that there is a perfect base pairing between these two strands. Now, the other thing that we learned from our ternary complex structure, that is in fact an inactive conformation, because if we look at the position of the catalytic center shown here by the star and the expected cleavage position shown by this dotted circle, or if we look at the schematic representation, so the catalytic center is roughly here and the cleavage position is located quite far away, it is clear that in order to perform cleavage at this distant position, the Fc domain must be somehow released or ejected from its original binding location. How does this happen? So by comparing the structure of the ternary and binary complexes of TnpB, we notice that in these complexes, the position of stem 1 is different. So in the binary complex, so the stem 1, shown here in cyan, it bends towards the core of the protein, towards the left, whereas in this ternary complex, it was not well resolved, but it is clear that it starts to bend away from the complex. And this bending is caused by formation of the heteroduplex here. If we have a short heteroduplex, it can fit without displacing stem 1, but as soon as the heteroduplex gets longer, say 12 or 15 base pairs, it starts clashing with this stem 1 and binary conformation, and it results in its displacement. So we believe that perhaps this bending of stem 1 may act as a trigger that activates Fc release and activation of TnpB. So indeed, we tested this experimentally by using a series of DNA substrates that support formation of various heteroduplexes of various length. And if we have short heteroduplexes, 6, 8, or 10, we observe no activity. In this case, we were testing so-called trans-cleavage and non-specific DNA cleavage activity of TnpB. But as soon as the heteroduplex length increases to 12 base pairs or longer, 14, 16, etc., we observe activation of this TnpB. It suggests that in this case, this Fc domain is released. So here we have a simple mechanism, schematic. So when the DNA is bound and the heteroduplex is formed, it displayed as stem 1, and this displacement acts as a trigger to release this green Fc domain. And this released Fc domain then starts cleaving the DNA, specific DNA, and the DNA in trans. The last thing not the last, but one other important thing that these structures revealed is that TnpB, in fact, represents the minimal structural core of the whole Cas12 family. So TnpB being perhaps the smallest member of this family, which binds quite a large and complex RNA. And if we have the direct opposite, it would be Cas12a, a well-known genome editor. It's a huge protein, 3 times larger than TnpB, but it has a very small RNA. But it has many insertions shown here in white. Presumably, all these extra protein elements that were acquired during evolution, they replaced them with these RNA elements present in TnpB. There are some cases that fit in between these two extreme cases. So, for example, we have a protein that is a group of Cas12 nucleases that are called Cas12F. These are a bit larger than TnpB, and unlike TnpB, they work as homodimers. So we have a second protein monomer shown here in dark blue. Like TnpB, it has quite a large and complex RNA bound. And in this case, this RNA is composed of two components of the CRISPR RNA, one of the trans-CRISPR RNA, similar to Cas9 enzymes. Now, for genome editing purposes, people usually connect these two RNAs via a short linker, hairpin loop, forming a so-called single guide RNA. Now, if you look at this stem 5 and this Cas12F scheme, and that stem 2 and TnpB, you can see that these are two equivalent. And in fact, TnpB already has this hairpin loop occurring naturally in its RNA. So in a way, nature discovered this single guide RNA first. Oh, another peculiar example of sort of intermediate properties of Cas12 nucleases is Cas12M enzyme shown here. So it's also a bit larger than TnpB and Cas12F, but it has a quite short and relatively simple RNA. An unusual feature of this enzyme is that it's not an enzyme, in fact, because though it has a C domain, it does not cleave DNA. And it performs its function by tight binding to its recognition site. It is sufficient to, let's say, repress viral replication or transcription. And it does by forming many contacts to this DNA. And some of these contacts are mediated by this extra element present in the cell of the elongated REC domain that we call roof. That sits on top. It forms many complexes with this non-target strand. Now, this brings me to the acknowledgement section. So we did most of the structural work together with Giedrė Tamulaitienė. He's our last speaker today. We did also collaborated with Artūras Karabis and Guillermo Montoya from the University of Copenhagen. Well, other people here, Gytis Tautvydas, Arūnas, and others, they helped with the design, and they have also conducted the biochemical research for this biochemical experiment. And I would also like to thank Eva Drolita from Thermo Fisher, who was our first instructor of Cryo-EM. So she taught us a lot. So thank you. And I will try to answer some of the questions. I do not see any questions. So, questions here. So if you have any other questions, I think you can type them in, and we will try to answer them afterwards or during the presentation. I think we can move to our next speaker, to Monika Jasnauskaitė from the EMBL Partnership Institute at the Vilnius University Life Sciences Center. And she will tell us about bacterial retrons. Thank you.
Hello, thank you for the introduction. My name is Monika, and today I will talk about my PhD work, which I'm doing at MBL Partnership Institute at Patrick Spausch Lab. My work focuses on structure and mechanisms of retron EC67. For a very long time, it was not known what retrons are and what they do. Retron EC67 was mentioned for the first time in the 90s. The authors identified an open reading frame, which sequence had resemblance to reverse transcriptase and RNase H. They showed that it produced a multi-copy single-stranded DNA, which was linked to RNA. They proposed a possible secondary structure. They showed that DNA and RNA could form stem loops, and there might be a DNA-RNA hybrid at the 3' end. Only in 2020, it was shown that retrons are defense systems. They provide bacteria defense against the phages. They consist of reverse transcriptase, non-coding RNA. They can have different effector proteins that are different for each retron system. Retron's non-coding RNA consists of two parts. One of them is, as it's labeled in the scheme in orange, is used for single-stranded DNA synthesis, and another part here in the scheme in red remains RNA in the final major retron complexes. And during the biogenesis, RT recognizes the conserved G base, which is used for initiation of single-stranded DNA synthesis. During this process, RNase H degrades the template for the synthesis. The synthesis stops at the 3' end, where it leaves the RNA-DNA hybrid at the 3' end. EC67 is the second most abundant retron system. It is unique and interesting because in contrast to other retron systems, it consists of single gene, where RT is fused to endonuclease, while others have most of others have additional single genes. In the study, the authors showed that EC67 protects against T5 phage. Another study besides that showed that it protects against T2 and T4 phages. They also specified that the endonuclease at the C-terminus is top rim, and they showed that the activity of both domains, RT and top rim, is essential for defense. Now we knew what the retron EC67 does. What is actually the mechanism of it? To answer this question, we determined its structure, determined its activator by screening phage libraries and identifying escapers. That allowed us to propose a possible activation mechanism. I'll start with the structure. We expressed and purified and prepared protein complexes for Cryo-EM. During this work, we wondered what the top rim domain might actually do. Previously, as I mentioned in the beginning, it was identified as RNase H, and we knew that RNase H is required for biogenesis to degrade the RNA template. So we hypothesized it might be required for biogenesis in EC67 case as well. Therefore, we thought if we create a complex mutant that has its top rim active site inactivated, that might interfere with single-stranded DNA production, and we might not get DNA synthesized. When we purified both the wild type and the top rim mutant complexes, we saw that they were identical. Here's the gel and the size exclusion chromatogram. We analyzed the DNA products that came with the complexes and saw that in both of the cases, the wild type and the top rim mutant, we had a DNA product produced. That suggested a top rim might not be involved in the biogenesis, but it could have a different function. Next, we determined the structure. We saw that EC67 forms heterohexamers. We have three fusion proteins of RT and top rim. Here on the top in light blue is RT. It looks like a flower. And we have three copies of RNA-DNA hybrid product. The RNA here is in orange. It's sitting in between the protomers. And the DNA is in dark blue. It's located on top of the RT and in the RT in the RNA-DNA hybrid. There's the top rim domain here at the bottom in yellow. We also did have some unexplained volume data, which can be seen here. We determined some presence of some nucleic acids, either RNA or DNA, which was going down from the RT domain onto the top rim domain here. Here's the close-up of the monomer. We have the RNA-DNA branch here. We have the RT active site here, Thoeris active site down at the bottom, and the RNA-DNA hybrid, which is located in between both of the active sites. And lastly, we saw some unidentified nucleic acid fragment, which was near the RT and the Thoeris domains. And we hypothesized it might be a secondary nucleic acid binding site. Now we knew how the complex looked like, therefore we asked how it is actually activated. For this, we screened basal phage collection and showed that EC67 protects against a range of phages. The families that we identified are displayed here. We were able to identify T2 phage escapers. A phage escaper is a phage that is still able to propagate even in the presence of a defense system. These phages have certain mutations in their genetic elements that allow them to escape the defense system. So if we identify those mutations, we can identify what triggers or activates our complex. All of the escapers had mutations in TnpB gene. TnpB is a DC-specific single-stranded DNase, which is hypothesized to degrade host DNA. TnpB and protein A, which is also a DNase, were identified by another group as well. Therefore, we had a complex that has a single-stranded DNA, and the activator of it is a single-stranded DNase. Could it be that the degradation of a single-stranded DNA activates our complex? So that brings me to the final part, a possible activation mechanism. We wanted to show activity, so we wanted to clone, express, and purify TnpB, but we faced a problem. TnpB is toxic and cannot be cloned. We asked whether it is possible to use some other DNase, for instance, DNase 1 as a proxy for TnpB. In such setting, if the DNase is degrading single-stranded DNA in the retron complex, and the retron complex is activated, it has RNase activity, it would cut the reporter, and we would be able to measure the fluorescence. And indeed, we saw that when the retron was treated with DNase, it showed an increased fluorescence signal. Here in this can be shown in this second bar in comparison to retron alone. Previously, we thought what Thoeris domain might do. We showed that it might not be necessarily involved in biogenesis. Therefore, we suggested maybe it could be the effector of the complex. So we joined this Thoeris mutant to our activity assay. And we saw that there was no increased fluorescence signal in comparison to wild type treated with DNase 1. And lastly, we had our background controls. That brought us another question, how this RNase activity could be regulated. We turned back to the structure and to this hypothetical secondary nucleic acid binding site, which was located near the RT in blue and Thoeris in yellow. We thought that upon phage infection and DNA degradation, this region might undergo through some structural changes that might lead to the activation of the Thoeris domain. We looked at some amino acids like these lysines here and this tyrosine, and suggested they might go through some changes. Another quite interesting feature which is near the site is the trimerization interface, this loop. It could be better seen from the bottom of the complex. So now the Thoeris domains are on the top and facing towards us. And here in the middle, we can see these loops that are coming from the RT domain. If we zoom in, we can see that in these interactions, tyrosines and valines are participating. We asked whether those amino acids that I've talked about here could be important. Therefore, we decided to mutate them and test the complex ability to defend bacteria from the phage. Here in this graph, the first bar represents the wild type. It shows that the defense system is functioning and the bacteria is protected. The second bar represents empty control, so no defense system and bacteria susceptible to infection. And the rest of the bars, they show these mutations of those amino acids that I've mentioned here. So we saw that they are indeed relevant, and we are currently working to investigate these interactions more. I would like to conclude with some of the most important findings that we had. EC67 forms a heterohexamer, which sandwiches protein RNA. This phage senses phage DNases. Single-stranded DNA degradation primes toperim for RNA degradation. Of course, some of the questions are still open. For instance, what's the substrate specificity? And we're working on this question at the moment. I would like to conclude by thanking the people who are involved in this work, my mentor Patrick Bausch, our student Kristina, people from Malinauskaitės lab, Lina and Algirdas, who helped us prepare samples for Cryo-EM and to process Cryo-EM data, and people from Virginijus Šikšnys lab, Inga and Toma for phage escaper generation, and Jonas for phage escaper sequencing. And of course, our funding agencies. I want to thank you for your attention. If you have any questions, I would be happy to answer them.
Okay, thank you, Monika. Yes, Q&A. I do not see questions here. So I do see one question in the chat that is directed to the first presentation. So perhaps I will answer to Jose Maria Carazo a bit later. But now, are there any? No question. Perhaps I can ask one. What would be the most the major substrate for this activated retron? You told us that you are still working on this topic, but what do you suspect? I mean,
yes, we would suspect the RNAs that are involved in protein translation processes. It was shown that some old family nucleases that have, for instance, C-terminus toperim domain, they also degrade some RNA substrates that are required for protein synthesis. So it might be a possible target for this retron.
Okay, thank you. Okay, we have some time, so perhaps I will ask another question. How do you, this peculiar structure of this retrons is that symmetric, highly symmetric. What is the purpose of the symmetry? Is it this sort of helps with this allosteric activation, or what are your ideas on this?
Yeah, it's a really good question. It's a question that we are also wrapping our heads about it. Yes, it's probably related to some biogenesis processes, how the DNA products are being produced and how this whole protein and nucleic acid complex would assemble. But probably from the evolutionary side, it was possibly the best option and the best complex composition to perform its function, because it's the second most abundant system among the retrons. So it should such symmetry and such assembly should have some evolutionary benefits.
Okay, thank you. We have one question in the Q&A from Arturo Carabias. Very beautiful structure and presentation. Is the MS DNA degraded during infection? DNA?
Yes, that's. I'm not able maybe to precisely tell what would be the how the event should go, but it should be like right after infection, so the complex could respond to the phage particle present, and it could lead to abortive infection to prevent virus particles from spreading. So I would think it should be during the infection.
Okay, thank you. So, thank you, Monika. I think let's move to our last speaker, Giedrė Tamulaitienė. So, please, Giedrė. So Giedrė will tell us about the bacterial antiviral defense system that is called Thoeris.
Okay, so hello, I'm a group leader in this Life Sciences Center, and I will talk about my project on Thoeris. So first of all, I would like to mention that bacteria possess over 100 different antiviral defense systems, and well, some of them share common mechanisms. So Thoeris belongs to so-called signaling systems. These systems are composed of two components. So one is a sensor that recognizes phage infection by different ways, and then it synthesizes signaling molecules, so second messenger, which usually is cyclic nucleotides. And then these cyclic nucleotides are bound by the second component, which is effector protein, and it usually has some enzymatic activity, which leads to cell death or dormancy. And in this way, this bacteria stops viral production and protects other cells from viruses. And the first signaling system was discovered in our department by Giedrės Tamulaitienės group. It is a type 3 CRISPR-Cas systems, which by recognition of phage transcript, it's type 3 effector complexes start synthesizing signaling molecules that are cyclic oligoadenylates that are bound by different effectors present next to these type 3 systems. And these effectors can have different enzymatic activities, like first effector CSM6, which has a heparin domain for unspecific RNA cleavage. And we also, together with Tamulaitienės group, characterized other different effectors like mRNA interferase or protease. And so Thoeris is also a signaling system. It was named by Egyptian goddess of childbirth and fertility. And it's quite common system. It's found in around 4% of sequenced bacterial and archaeal genomes. So this system is rather simple. It is composed of two genes, THSA and B. And the THSA is an effector. It has SIR2-like SIR2 domain and nucleotide binding slug domain. And THSB is a sensor. It comprises TIR domain, so tall interleukin-1 receptor, that is also common in eukaryotic immunity systems. So the best characterized system of Thoeris is from Bacillus cereus. And when we started this our studies, so it was already known that THSB recognizes phage infection and produces signaling molecule, so is a cyclic ADPR isomer called glycocyclic ADPR. And then this glycocyclic ADPR is bound by the slug domain of THSA effector and induces NAG cleavage by its SIR2 domain. And however, this already apostructures of these proteins was known, so THSB was a monomer, and it was inactive in vitro without the phage clue. And THSA was a tetramer in crystal and solution. So we decided to study how this protein is activated. And next to this well-characterized operon of Thoeris, we found another third domain protein we call THSB prime. And we purified the protein, and we were lucky to find that it is active in vitro. So it demonstrated quite low but significant NAD cyclization activity. And this produced the glycocyclic ADPR. It was able to activate NAD cleavage activity of THSA, what we see with all NADs cleaved when both proteins are present. And we also could measure its cleavage by the fluorescent assay. So that's why we were able to reconstitute Thoeris activity in vitro. And by DLS, we found that binding of glycocyclic ADPR induces oligomerization of THSA protein. So how does oligomer look like? We used Cryo-EM to determine it. So Cryo-EM grids already showed very long filaments formed by activated THSA and very nice 2D classes. And we were able to reconstruct helical filaments of THSA. And we found that helical filament is composed of THSA tetramers, shown here in different colors. And the helical core was formed by SIR2 domains, colored in cyan, and slug domain dimers were sticking away from this helix. When we looked at these domain structures, so we found a glycocyclic ATPR molecule bound in the slug domain pocket of the activated protein and made numerous contacts with the protein. And when we compare slug domain structures with activated and apo forms, so activated in yellow and apo in gray, we see that very small conformational differences in the loop regions only could be observed, and it was quite surprising for us. But when we looked at SIR2 domains, we found that SIR2 domains show much larger conformational differences. So the conserved-Rossmann fold core was superimposed quite well in apo and activated structures. However, in the we observed larger conformational differences in these helical regions. And in the active site of SIR2 domain, we found part of NAD molecule shown here in yellow. And it superimposed quite well with Archaeoglobus fulgidus, SIR2 homologue of THSA. And this NAD fragment is also shown here, and we can see that in the apo structure, this red loop blocks the active site of THSA, and it is removed into different conformation in this helical structure. When we look at the tetramer structure, so in the apo tetramer is asymmetric, and it's not flat. And when it's activated and bound with this glycocyclic ADPI, it forms very nice symmetrical tetramer that can be already form a helical filament. So our findings found completed almost mechanism of type 1 Thoeris. So after cognition of phage signal, THSB forms. THSB makes glycocyclic ADPR from NAD, and it is bound by the asymmetric THSA, and in the slug domain induces its conformational changes and then further polymerization of the tetramers, and these helical interactions stabilize the active site of SIR2 domain and allows rapid depletion of NAD and cell death. And recently it was published that THSB is activated by a viral major capsid protein. However, this structural mechanism of this activation is not known yet. And also, viruses found a way to escape Thoeris protections, so they produce Thoeris anti-defense proteins, or TATs, that could bind the glycocyclic ADPI signaling molecule like as sponges. So two types of Thoeris anti-defense proteins are known. TAT1 is a dimer, binds two GC-ADPI molecules, and TAT2 is a tetramer, and also binds two GC-ADPI molecules. And well, I talked about type 1 about one Thoeris system, which is called type 1. And the original paper described two variants of Thoeris. So another type of Thoeris, what we call type 2, has also a sensor protein composed of TIR domain, but it has different effector protein, which is composed of transmembrane and macrodomains. So macrodomain is found in all kingdoms of life, and it's known for binding ADPR in free form or bound to proteins and nucleic acids. And some of macrodomains have also ability to remove ADPR modifications from proteins and from nucleic acids. So the question remains, what signaling molecule? Is it the same for both types of Thoeris, or is it different? So to answer this question, we, together with collaborators from the CEREC lab, performed chimeric variants of type 1 and type 2 systems. So we took two type 1 systems and one type 2 system, and we see that when we have both proteins from the same types of systems, we have protection against phages. However, if we mix one B and A proteins from different systems, we observe that we lose this protection against the phages. So this suggests us that signaling molecules are different. We also, our collaborators also found anti-Thoeris proteins variant that protects only, that inhibits only type 2 Thoeris, but has no effect on type 1 Thoeris, also further supporting our hypothesis on signaling molecule. And collaborators from Giedrius lab determined crystal structure of this MOT-TAT2 variant specific for type 2, and found that it differently from type 1, it had 4 potential binding pockets. So we used the MOT-TAT2 to pull down the signaling molecule. So bacillus cells expressing only THSB protein of type 2 were infected with the virus. And after a certain time of infection, the cell lyses that were collected and filtrated through 3 kilodalton filter to collect only small molecules. And this lysate was incubated with the purified anti-Thoeris protein. And then this protein was concentrated and washed away and further boiled to remove protein and just have this pull down signaling molecule. And in MS, we see that we had some peak after infection, and then the same molecule was pulled down with MOT-TAT2. And when we determine mass of this molecule, we see that it is higher than mass of NAD, which is a typical substrate for TIR domain proteins. So we have some conjugates with NAD probably. And last, we determined the crystal structure of soluble THSA macrodomain, which was co-expressed with its cognate THSB protein. And we see that this macrodomain is mostly similar to macro D type, and we here can see our structure in blue and homologs in yellow and green. And when we compare binding pockets of these macrodomains, so we see that this site that binds adenine is quite conserved, and while this part which is located with the distal ribose, it's a bit different in all proteins. And when we examine the pocket, we found that it contains bound molecule, and this electron density allowed us, and contact with the protein allowed us to identify that we have histidine ADPR conjugate. So it's first signaling molecule that is found of nucleotide bound to amino acids. And in this, histidine is bound to ADPR through its imidazole tau atom, and it makes a beta-glycosidic bond, and which is different from alpha-glycosidic bond, which is found in ADP ribosylated proteins. And this theoretical mass of histidine ADPR perfectly matches our value of a known molecule, which is obtained in this in vivo experiments. Histidine ADPR makes a lot of contacts with the proteins, so about 30 direct and water-mediated hydrogen bonds to the protein. And when we mutated amino acid residues that are involved in adenine or histidine binding, here we see that we lose protection of Thoeris against the phages. So most of these residues are important, so like without Thoeris system. And how it could protect, we don't have a post structure, so we used AlphaFold model to induce protection, and we find that while monomer structure overlaps perfectly, so we do see difference in the dimer formation. So probably binding of his ADPR could induce some dimer rearrangement, and we used AlphaFold to make structures of full-length protein in the dimer mode. So, and also if we ask for oligomer, like 6 THC molecules, we have some AlphaFold model with oligomer protein. So probably oligomerization through these macrodomains is formed by binding of his ADPR, and this oligomer in the membrane somehow disrupt the membrane. And the similar mechanism was suggested for another signaling system, CBAS, with the transmembrane effector. And recently, type 4 structure of type 4 mechanism was proposed. And it's interesting that in all systems signaling, well, sensor proteins are THSBs with TIR domains, but they produce 3 different signaling molecules. So first, type 1 produces glycocyclic ADPR, type 2 histidine ADPR, and type 4 produces already novel type of cyclic ADPR, so N7 cyclic ADPR. So typical ADPR is cyclic ADPR is N3 cyclic ADPR, and use different effectors, so like NAD depletion, membrane disruption, or protein degradation by type 4 to protect bacteria against phages. And finally, I would like to thank people who were involved. So for my group, so done mostly by my PhD student Džiugas Sabonis, and also Davidas Giedrius, Arūnas Audronė, Mindaugas Virgis, and also from Rotem Sore group, mostly by Carmel Abraham, and from Philip Kranzl group with Alan René. So thank you for your attention, and I'm ready to take questions.
Thank you, Giedrė. I'm looking at the Q&A. No questions here. Perhaps I can ask you a question. Now we have different signaling molecules. The last one, which is the most recent, published with this N7, it looked quite symmetric in a way due to these covalent bonds to this purine. Is it bound in some symmetric environment? Is it known what is the binding site for this new molecule?
No, it is still unknown, just no structural data on this system yet. So it's just biochemical data and, well, in vivo data showing that it's somehow activated.
Okay, thank you. Okay. More questions? Well, I still have unanswered questions by Jose Maria Carazo from University of Madrid that was directed to me. So it is related that on the preferred particle orientation. So it reads, you were finally able to go around the preferred orientation of the single and DNA substrate, but did you also try tilting? Also, depending on how serious is the case, the isonet-like algorithm by Hongzhou may provide some clues. So in our case with TnpB, we did try tilting, and it helped a bit. The issue here, but it was not as good as these do to end substrates. Or maybe we tried, in fact, both approaches simultaneously. The issue here is that these molecules are quite small, around 100 kilodaltons, and perhaps tilting sort of makes the quality of the data somewhat worse. And we did not use a SizeNet-like algorithm, but I do presume that it might help us, but only with this improved data. The starting data was virtually single orientation, very limited information there. Yeah, so I hope that answers the question. Yeah, so thank you for participating in the webinar. Thank you for the questions and for nice presentations. And I hope we will meet you in some other webinars or conferences. Thank you.